Good day, this is Professor Will C. BMD, PhD. Today is Tuesday, December 4th, 2018. It is 21:25 and 8 seconds Eastern Standard Time. Science research. Research increasingly links the gut microbiome to a range of human maladies, including inflammatory bowel disease, diabetes, and even cancer. Attempts to manipulate the gut with food rich in healthy bacteria such as yogurt or kombucha are in vogue, along with buying commercial probiotics that promise to improve users' chances against illness. Changing the gut microbiome to beat illness really does hold great potential. But first, scientists must answer what constitutes a healthy gut microbiome in womb. By studying data on nearly 17,000 Americans of varying genders, ages, weights, and ethnicities, they learned that gut microbiome differences among ethnicities are the most consistent factor. That discovery holds promise in the burgeoning field of individualized medicine, because it is far easier to change a person's microbiome than their genes. The other major markers for disease: in addition, many chronic diseases disproportionately affect ethnic minorities with underlying causes of the disease unexplained. Perhaps some answers lie in that gut microbiome. Human genomes are 99.9% the same between any two people, so what we are really interested in is what explains the marked variations in gut microbiomes between people. What are the rules, and can we manipulate that microbiome in order to improve health and medicine in the long run? If you look at common factors associated with gut microbiome differences, such as gender, weight, or age, you find many inconsistencies in the same type of gut bacteria present. But when we compare differences between patients' self-declared ethnicities, we find stable and consistent features of bacteria present in the gut. The team discovered 12 particular types of bacteria that regularly vary in abundance by ethnicity. Because ethnicity captures many factors ranging from diet to genetics, it is difficult to say why this is, but it is a baseline for understanding healthy microbiome differences among individuals. You may buy probiotics over the counter at a drugstore, but those are unlikely to affect your microbiome in a substantiated way. Moreover, one size may not fit all. But with more of this kind of research, we can hone in on the relevant differences in doses of bacteria that may reverse illness or prevent it from developing in the first place. For today's is Professor Will C. BMD, PhD. Today is Wednesday, December 5th, 2018. It is 8:13:51 seconds Eastern Standard Time. Osteoarthritis, a disease that causes severe joint pain, affects more than 20 million people in the United States. Some drug treatments can help alleviate the pain. There are no treatments that can reverse or slow the cartilage breakdown associated with the disease. In advance, that could improve the treatment options available for osteoarthritis. MIT engineers have designed a new material that can administer drugs directly to the cartilage. The material can penetrate deep into the cartilage, delivering drugs that could potentially heal damaged tissue. This is a way to get directly to the cells that are experiencing the damage and introduce different kinds of therapeutics that might change their behavior. In a study in rats, the researchers showed that delivering an experimental drug called insulin-like growth factor 1, IGF-1, with this new material prevented cartilage breakdown much more effectively than injecting the drug into the joint on its own. Osteoarthritis is a progressive disease that can be caused by traumatic injury such as tearing a ligament. It can also result from gradual wearing down of cartilage as people age. A smooth connective tissue that protects the joints. Cartilage is produced by cells called chondrocytes, but is not easily replaced once it is damaged. Previous studies have shown that IGF-1 can help regenerate cartilage in animals. However, many osteoarthritis drugs that showed promise in animal studies have not performed well in clinical trials. The MIT team suspected that this was because the drugs were cleared from the joint before they could reach the deep layer of chondrocytes that they were intended to target. To overcome that, they set out to design a material that could penetrate all the way through the cartilage. The spear-shaped molecule they came up with contains many branch structures called dendromeres. That branch from a central core, the molecule has a positive charge at the tip of each of its branches, which helps it bind to a negatively charged cartilage. Some of those charges can be replaced with a short, flexible, water-loving polymer, known as PEG, that can swing around on the surface and partially cover the positive charge. Molecules of IGF-1 are also attached to the surface. When these particles are injected into a joint, they coat the surface of the cartilage and then begin diffusing through it. This is easier for them to do than it is for free IGF-1 because the Spirit's positive charge allows them to bind to cartilage and prevent them from being washed away. The charged molecules do not adhere permanently, however, thanks to the flexible PEG chains on the surface that cover and uncover charge as they move. The molecules can briefly detach from cartilage, enabling them to move deeper into the tissue. We found an optimal charge range so that the material can both bind the tissue and unbind the further diffusion, and not be so strong that it just gets stuck at the surface. Once the particles reach the chondrocytes, the IGF-1 molecules bind to receptors on the cell surface and stimulate the cells to start producing proteoglycans, the building blocks of cartilage and other connective tissues. The IGF-1 also promotes cell growth and prevents cell death. When the researchers injected the particles into the knee joints of rats, they found that the material had a half-life of about 4 days, which is 10 times longer than IGF-1 injected on its own. The drug concentration in the joints remained high enough to have a therapeutical effect for about 30 days. If this holds true for humans, patients could benefit greatly from joints injections, which can only be given monthly or bi-weekly, the researchers say. In the animal studies, the researchers found that cartilage in injured joints treated with the nanoparticle drug combination was far less damaged than cartilage in untreated joints or joints treated with IGF-1 alone. The joints also showed reductions in joint inflammation and bone spur formation. Cartilage in rat joints is about 100 microns thick, but the researchers also showed that their particles could penetrate chunks of cartilage up to 1 millimeter, the thickness of cartilage in a human joint. That is a very hard thing to do. Drugs typically will get cleared before they are able to move through much of the cartilage. When you start to think about translating this technology from studies in rats to larger animals and someday humans, the ability of this technology to succeed depends on the ability to work in thicker cartilage. The researchers began developing this material as a way to treat osteoarthritis that arises after traumatic injury, but they believe it could also be adapted to treat age-related osteoarthritis. We now plan to explore the possibility of delivering different types of drugs, such as other growth factors, drugs that block inflammatory cytokines, and nucleic acids such as DNA and RNA. Today's is Professor Will, CBMD, PhD. Today is Wednesday, December 5, 2018. It is 8:58:48 seconds Eastern Standard Time. New quantum materials could take computing devices beyond the semiconductor era. Multi-Farogs are promising candidates for new type of memory and logic circuits. Date December 3, 2018. Source University, California, Berkeley. Summary: scientists in industry and academia are looking for new materials to succeed highly successful semiconductor transistors. Scientists have hit on a very promising post-transistor technology, multi-Farogs, which use magnetic spin states instead of electron charge to store binary data. They have shown that these MESO, magnetic electric spin orbit, devices can greatly improve energy efficiency and pack more logic devices onto a chip. Single crystals of the multi-Farog material bismuth iron oxide. The bismuth atoms blue form a cubic lattice with oxygen atoms yellow at each face of the cube and an iron atom gray near the center. The somewhat off-center iron interacts with the oxygen to form an electric dipole P, which is coupled to the magnetic spins of the atoms M, so that flipping the dipole with an electric field E also flips the magnetic moment. The collective magnetic spins of the atoms in the material encode the binary bits 0 and 1, and allow for information storage and logic operations. In a paper appearing online December 3rd in Advance in publication in the Journal of Nature, the researchers proposed a way to turn relatively new types of materials, multi-Farogs and topological materials, into logic and memory devices that will be 10 to 100 times more energy efficient and foreseeable improvements to current microprocessors, which are based on CMOS complementary metal oxide semiconductor. The magnetoelectric spin, orbit or meso devices will also pack 5 times more logic operations into the same space as CMOS, continuing the trend toward more computations per unit area, a central tenet in Moore's law. The new devices will boost technologies that require intense computing power and low energy use, specifically higher automated self-driving cars and drones, both of which require ever-increasing numbers of computer operations per second. As CMOS develops into a maturity, we will basically have very powerful technology options that see us through. In some ways, this could continue computing improvements for another whole generation of people. Transistor technology invented 70 years ago is used today in everything from cell phones and appliances to cars and supercomputers. Transistors shuffle electronics around inside a semiconductor and store them as binary bits 0 and 1. In the new meso devices, the binary bits are the ups and down magnetic spin states in multi-Farad, a material first created in 2001. The discovery was that there are materials where you can apply a voltage and change the magnetic order of a multi-Farad. Researchers report that they have reduced the voltage needed for a multi-Farad magnetic electric switching from 3 volts to 500 millivolts and predict that it should be possible to reduce this to 100 millivolts, 1/5 to 1/10 that required by CMOS transistors in use today. Lower voltage means lower energy use. The total energy to switch a bit from 1 to 0 would be 1/10 to 1/13 of the energy required for CMOS. A number of critical techniques needed to be developed to allow these new types of computing devices and architectures. Scientists who combine the functions of magnetoelectrics and spin-orbit materials to propose MESO. We are trying to trigger a wave of innovation in industry and academia on what the next transistor-like option should look like. Internet of Things and AI. The need for more energy-efficient computers is urgent. The Department of Energy projects that with the computer chip industry expected to expand to several $1 trillion in the next few decades, energy use by computers could skyrocket from 3% of all U.S. energy consumption today to 20%, nearly as much as today's transportation sector. Without more energy-efficient transistors, the incorporation of computers into everything, the so-called Internet of Things, would be hampered, and without new science and technology, scientists lead in making computer chips could be unstaged by semiconductor manufacturers in other countries. Because of machine learning, artificial intelligence, and IoT, the future home, the future car, the future manufacturing capability is going to look very different. If we use existing technologies and make no more discoveries, energy consumption is going to be large. We need new science-based breakthroughs. Multi-Farads and spin-orbit materials. Multi-Farads are materials whose atoms exhibit more than one collective state. In ferromagnets, for example, the magnetic moments of all iron atoms in the material are aligned to generate a permanent magnet. In ferroelectric materials, on the other hand, the positive and negative charges of atoms are offset, creating electric dipoles that align throughout the material and create a permanent electric moment. MESO is based on multi-Farad material consisting of bismuth iron and oxygen BiFeO3 that is both magnetic and ferroelectric. Its key advantage is that these two states, magnetic and ferroelectric, are linked or coupled so that changing one affects the other. By manipulating the electric field, you can change the magnetic state, which is critical to MESO. The key breakthrough came with the rapid development of topological materials with spin-orbit effect, which allow for the state of multi-Farad to be read out efficiently. In MESO devices, an electric field alters or flips the dipole electric field throughout the material, which alters or flips the electron spins and generate the magnetic field. The capability comes from spin-orbit coupling, a quantum effect in materials which produces a current determined by electron spin direction. Another paper that appeared earlier this month in Science Advances, UC Berkeley and Intel experimentally demonstrated voltage-controlled magnetic switching using the magnetic electric material bismuth iron oxide BiFeO3, a key requirement for MESO. We are looking for revolutionary and evolutionary approaches for computing in the beyond CMOS era. MESO is built around low-voltage interconnects and low-voltage magnetoelectrics, and brings innovation in quantum materials to computing. Good day, this is Professor Will, CBMD, PhD. Today is Wednesday, December 5th, 2018. It is 20:32 and 55 seconds Eastern Standard Time. Science News. Early clinical trial data show gene therapy reversing sickle cell anemia. Treatment uses reduced intensity preconditioning to enhance its global transportability. Date December 4, 2018. Source Cincinnati Children's Hospital Medical Center. Summary. After over a decade of preclinical research and development of new gene therapy, treatment for sickle cell anemia, SCA, is reversing disease symptoms in 2 adults and showing early potential for transportability to resource-challenged parts of the world where SCA is most common. After over a decade of preclinical research and development A new gene therapy treatment for sickle cell anemia, SCA, is reversing disease symptoms in 2 adults and showing early potential for transportability to resource-challenged parts of the world where SCA is most common. Preliminary data from pilot phase 1-2 clinical trial testing of gene addition therapy were presented December 2nd at the American Society of Hematology ASH annual meeting in San Diego by principal investigator. 1 year after treatment of our first patient and 6 months after treatment of our second patient, both have seen a remarkable improvement in the quality of life due to a remarkable reduction in disease symptoms. This includes near elimination of chronic pain and sickling events and improved anemia. Although it is still early post-treatment, these preliminary results are quite promising. If sustained, this therapy will provide a transportable, safe, and feasible gene therapy for all SCA patients. SCA is a painful inherited disease where blood cells become sickle-shaped and stuck in the vascular system. It could damage vital organs and cause death. How the therapy works. The gene therapy developed by Cincinnati Children's uses a modified gamma-globin lentivirus vector to transfer healthy fetal hemoglobin gene into a patient's blood stem cell, hematopoietic stem cells, HSC. The lentivirus is modified so it does not cause illness. The therapy comes from the observation that some adults never stop producing fetal hemoglobin, HbF. This prevents red blood cell sickling when sufficient quantities of HbF are present in the body. Normally, the fetal hemoglobin gene switches off shortly after birth. The new gene therapy approach designed by researchers and team places a fetal hemoglobin gene in the bone marrow cells that cannot switch off. Cells are collected earlier from the SCA patient and genetically reprogrammed in the clean lab facility. With the modified lentivirus vector, then the patient bone marrow is preconditioned with 1 low dose of chemotherapy, after which gene-corrected cells are infused back into the patient. Patients recover their blood counts within a week to 10 days and recover from a chemotherapy effects by 2 weeks. A key feature of the treatment regimen and the reduced intensity of preconditioning for patients receiving the therapy for SCA. The method is less toxic and expensive than myeloablative preconditioning normally used in bone marrow transplants, according to the researchers. Myeloablative conditioning uses high-intensity chemotherapy to prepare transplant patients' bone marrow for healthy gene-corrected blood stem cells, HSC. This typically results in low blood counts and other side effects. These can include severe infections and long stays in transplant units for 1 or 2 months. This means transplants using myeloablative conditioning have to be performed at large and sophisticated medical centers in developed parts of the world. Researchers say this limits access to people in underdeveloped regions where SCA is more common, like Africa, India, the Caribbean, and South America. Reaching high-need regions. One goal for researchers and colleagues is that their therapy, reduced intensity conditioning, will allow it to be offered at hospitals in places like Central Africa and other underdeveloped regions of the world. SCA affects more than 90,000 people in the United States, and millions of people worldwide have the disease, according to the researchers. Researchers say additional data from the study will tell investigators of the consistent and long-term clinical benefits of the therapy for patients with severe SCA. The trial is scheduled to treat up to 10 patients with SCA. Preclinical studies by the research team suggested the treatment allows the body to produce normal red blood cells instead of the sickle-shaped cells that define the inherited disorder. Improved quality of life. Both adult patients included in the ASH presentation are 35 to 25 years old who, respectively, had gene transfer therapy 15 and 12 months ago. In the year and a half prior to receiving gene correction therapy, both patients had more than a dozen acute sickle events per year, according to researchers. These included acute chest and pain crisis, chronic pain, and required the ongoing use of oral and injectable opioids on a regular basis. Investigators report that 1 year following therapy, the first patient no longer requires daily oral opioids for chronic pain and has had 1 acute sickle cell event. The second patient no longer uses oral opioids and has no vaso-occlusive events, where blood cells become stuck in the vascular system. Good morning, this is Professor Will, CBMD, PhD. It's Thursday, December 6, 2018. It is 6:25, 38 seconds Eastern Standard Time. Science News. Migraines that affect vision may increase risk of irregular heartbeat. Date November 14, 2018. Source American Academy of Neurology. Summary. People who experience migraine with visual aura may have an increased risk of irregular heartbeat called atrial fibrillation, according to new study. People who experience migraine with visual aura may have increased risk of an irregular heartbeat called atrial fibrillation, according to a study published November 14, 2018. Online issue of Neurology, the Medical Journal of American Academy of Neurology. Migraine with visual aura is when disturbances in vision occur right before head pain begins. Those disturbances may include seeing wavy lines or flashes of light, or having blurry vision, or blind spots. With atrial fibrillation, a form of arrhythmia, the heart's normal rhythm is out of sync. As a result, blood may pool in the heart, possibly forming clots that may go to the brain, causing a stroke. Since atrial fibrillation is a common source of strokes caused by blood clots, and previous research has shown a link between migraine with aura and stroke, we wanted to see if people who have migraine with aura also have a higher rate of atrial fibrillation. Atrial fibrillation can be managed through medicine, but many people do not realize that they have atrial fibrillation. For the study, 11,939 people with an average age of 60 without prior atrial fibrillation of stroke were evaluated for headache. Of those, 9,405 did not have headache, and 1,516 had migraine. Of those who had migraine, 426 had migraine with visual aura. The participants were followed for up to 20 years. During the study, 1,623 people without headache, or 17%, developed atrial fibrillation, while 80 or 440 people with migraine with aura, or 18%, developed the condition, and 152 and 1,105 people with migraine without aura, or 14%. After adjusting for age, sex, blood pressure, smoking, and other factors that could affect the risk of atrial fibrillation, people with migraine with aura were found to be 30% more likely to develop the condition than people who did not have headaches, and 40% more likely to develop atrial fibrillation than people with migraine with no aura. The results translate to an estimated 9 out of 1,000 people with migraine with aura having atrial fibrillation compared to 7 out of 1,000 people with migraine without aura. Researchers also found that the rate of stroke in the migraine with aura group was 4 out of 1,000 people annually, compared to 2 out of 1,000 people annually in those with migraine without aura, and 3 out of 1,000 people annually in those with no headache. Our research suggests that atrial fibrillation may play a role in stroke in those with migraine with visual aura. It is important to note that people with migraine with aura may be at higher risk of atrial fibrillation due to problems with the autonomic nervous system, which helps control the heart and blood vessels. More research is needed to determine if people with migraine with visual aura should be screened for atrial fibrillation. A limitation of the study was that the definition of migraine may have left out people who had migraines that lasted less than 1 year or who had a history of migraine at younger ages. There was also limited information on migraine medications that may influence heart rate. For data, this is Professor Will CBMD PhD. Today's Thursday, December 6, 2018. It's 21:28:30 Eastern Standard Time. Science News. Natural compound 2HF treats leishmaniasis infection study finds. Date December 6, 2018. Source Plus. Summary: current treatment options for the parasitic disease leishmaniasis are largely ineffective, inexpensive, and tend to be plagued by resistant parasites and side effects. Now researchers have showed that natural flavonoid is effective at treating Leishmania amazonis infections. Leishmaniasis is endemic to 98 countries and affects more than 12 million people around the world. Treatment is possible with pentavalent antimonial, but many Leishmania parasites have become resistant to antimonial chemotherapy, flavonoids, a group of natural compounds that are isolated from fruits, vegetables, wine, and coffee. Have been considered a promising candidate for leishmaniasis treatment. 2-hydroxyflavonone, 2HF, part of a class of flavonoids found in citrus fruits, is already being studied for its use in cancer chemotherapy. Researchers studied the effect of 2HF on wild-type and antimony-resistant L. amazonius parasite cells, and then went on to test the effectiveness of the compound in treating mice with L. amazonius infections. 2HF stopped the proliferation of leishmaniasis amazonius parasites in both their free-living form and the form that infects human cells in both strains of Leishmaniasis amazonius tested. In addition, 2HF reduced the overall number of parasites in the affected mouse and reduced the size of lesions caused by the cutaneous form of leishmaniasis. No side effects were seen in the treated mice. Considering that 2HF reduced the lesion size and parasite load without compromising the overall health of the infected mice, we suggest this compound is a potential candidate for leishmaniasis chemotherapy for cutaneous leishmaniasis. The researchers say studies should be conducted to determine the ideal dose and therapeutical regime. Today, this is Professor Will, CBMD, PhD. Today is Thursday, December 6, 2018. It is 22:06 and 43 seconds Eastern Standard Time. Sensors developed to detect and measure cancer's ability to spread. Date, December 5, 2018. Source, University of California, San Diego. Summary. Researchers engineered sensors to detect and measure the metastatic potential of a single cancer cells. Metastasis is attributed as a leading cause of death in people with cancer. The spread of invasive cancer cells from a tumor's original site to distinct parts of the body is known as metastasis. It is the leading cause of death in people with cancer. In a paper published online in iScience, University of California, San Diego School of Medicine, researchers reported engineering sensors that can detect and measure the metastatic potential of single cancer cells. Cancer would not be so devastating if it did not metastasize. Although there are many ways to detect metastasis once it has occurred, there has been nothing available to see or measure the potential of a tumor cell to metastasize in the future. So at the Center for Network Medicine, we tackled this challenge by engineering biosensors designed to monitor not only one, not two, but multiple signaling programs that drive tumor metastasis. Upon sensing those signals, a fluorescent signal would be turned on only when tumor cells acquired high potential to metastasize and therefore turn deadly. Cancer cells alter normal cell communications by hijacking one of many signaling pathways to permit metastasis to occur. As the tumor cells adapt to the environment or cancer treatment, predicting which pathway will be used becomes difficult. By comparing proteins and protein modifications in normal versus all cancer tissues, researchers identified a particular protein and its unique modification called tyrosine phosphatase CCD-C88A GIV/GIRDIN that are only present in solid tumor cells. Comparative analysis indicated that this modification can represent a point of convergence of multiple signaling pathways commonly hijacked by tumor cells during metastasis. The team used novel engineered biosensors and sophisticated microscopes to monitor the modification on GIV and found that indeed fluorescent signals reflected a tumor cell's metastatic tendency. They were then able to measure the metastatic potential of single cancer cells and account for the unknowns of involving tumor biology through this activity. The result was the development of fluorescent resonance energy transfer, FRET, abbreviated FRET, biosensors. Although highly aggressive and adaptive, very few cancer cells metastasize, and that metastatic potential comes and goes. If metastasis can be predicted, this data could be used to personalize treatment to individual patients. For example, patients whose cancer is not predicted to metastasize or whose disease could be excised surgically might be spared from highly toxic therapies. Patients whose cancer is predicted to spread aggressively might be treated with precision medicine to target the metastatic cells. It is like looking at a magic 8-ball, but with a proper yardstick to measure the immeasurable and predict outcomes. We have the potential not only to obtain information on single cell level, but also to see the plasticity of the process occurring at a single cell. This kind of imaging can be used when we are delivering treatment to see how individual cells are responding. The sensors need further refinement, researchers and authors wrote. Good day, this is Professor Will, CBMD, PhD. Today's Thursday, December 6, 2018. It is 22:29 and 58 seconds Eastern Standard Time. Scientists design a way to track steps of cells development. New tool has potential to boost regenerative medicine, cancer research. Date, December 5, 2018. Source, Washington University School of Medicine. Summary, scientists have developed a new tool described as a flight data recorder for developing cells. Illuminating the path cells take as they progress from one type to another. The cellular tracking device could one day help scientists guide cells along the right paths to regenerate certain tissues or organs, or help researchers understand the wrong turn some cells might take on their way to becoming cancerous. Scientists hope to one day be able to take skin cells from a patient who needs a liver transplant, for example, and guide the skin cells along a known path that will result in a new liver. Without this cellular tracking device, researchers are able to study the original cells and the final cells in great detail. But the pathways cells take to reach their destinations have been largely unknown. There is a lot of interest in the potential of regenerative medicine, growing tissues and organs in labs to test new drugs, for example, or for transplants one day. But we need to understand how the reprogramming process works. We want to know if the process for converting skin cells to heart cells is the same as for liver cells or brain cells. What are the special conditions necessary to turn one cell type into any other cell type? We designed this tool to help answer these questions. According to the researchers, the tool could reveal cellular reprogramming, routes that might involve reverting skin cells back to different types of stem cells that could then mature into a new liver or other vital organ. Among many potential uses, the tool also could be applied in cancer research, recording the wrong turns normal cells might take to develop into tumors. Using their flight data recorder, the researchers performed experiments that uncovered some surprising details about the specific routes taken by cells that successfully completed their flight paths. The new research information may help researchers identify the best pre-flight conditions to expose the cells to in order to increase the likelihood that their journeys will be successful. The scientists want to file the right flight plan, so to speak, so all the cells arrive at the correct destinations. Right now, cell reprogramming is really inefficient. When you take one cell population, such as skin cells, and turn it into a different cell population, say intestinal cells, only about 1% of cells successfully reprogram. And because it is such a rare event, scientists have thought it is likely to be a random process. There is some correct set of steps that a few cells randomly hit upon. We found the exact opposite. Our technology lets us see that if a cell starts down the right path, reprogramming very early in the process, all of its related sibling cells and their descendants are on the same page, doing the same thing. The technique harnesses the natural properties of a virus that inserts tiny DNA barcodes called cell tags into each cell. As the cells divide, their unique barcodes are passed down to all the descendant cells. At several set time points during the 28-day cellular reprogramming window, new barcodes are added and sample of the cells are analyzed to see what they are doing at that waypoint. The cell tagging technique keeps track of which cells share common ancestors and how far back that common ancestor is found in the lineage. Like a family tree, indeed, beyond single flight tracking, the tool lets scientists build complex family trees of cells, where successfully reprogrammed cells could be traced all the way back to their early ancestors. Researchers said their study suggests that the state the cell is in at the time it receives the instructions to pre-program has already set the stage for whether it will be successful or not. This contradicts assumptions that a cell goes in many different directions when it is first instructed to reprogram. If we can unlock that cocktail of initial conditions that prime cells to be successful in their reprogramming, we can transform cells into the type we want with much higher efficiency. We'd like to reach 100% efficiency. That would be really exciting for the field of regenerative medicine. The researchers already may have identified one ingredient in the cocktail. They found that if a certain gene called METTL7A1 was turned on in cells, they were 3 times as likely to successfully reprogram compared with cells in which this gene is inactive. According to Morris, another interesting finding was that the cells that were not successful in their reprogramming didn't just end up all over the map. They appeared to converge at the same dead end, tending to revert back to look like the original cell type. We see a single dead end right now, but as technology improves, we think there is a possibility we will see multiple outcomes. The researchers studied mouse skin cells, exposing them to a molecular cocktail that triggers the DNA for the cells to open up, turn on new genes, and reprogram skin cells into another cell type called induced endoderm progenitors. Such cells give rise to both liver cells and the cells that make up the small intestine. In one example of potential application, research wants to grow mini guts to help the study of premature infants with a condition called short bowel syndrome. Babies born too early are at high risk of a disease that results in the death of tissue in the intestine. To stop the damage, some babies require surgery to remove that portion of the bowel, resulting in a shorter bowel. Since part of the intestine has been removed, these infants lose their ability to absorb nutrients. If we eventually can grow replacement intestine from the human version of these cells, that would be an amazing advance. Today's is Professor Will, CBMD, PhD. Today's Thursday, December 6, 2018. It's 23:07, 37 seconds Eastern Standard Time. Science News. Sea invertebrate sheds light on evolution of human blood and immune systems. Date December 5, 2018. Source Stanford Medicine. Summary. A marine invertebrate that lives underwater, colonies resembling fuzzy pinheads clinging to rocks, has a blood-forming system with uncanny similarities to that of humans, according to scientists. Botrytis scosiri, a marine invertebrate that lives in underwater colonies resembling fuzzy pinheads clinging to rocks, has a blood-forming system with uncanny similarities to that of humans, according to scientists at Stanford University. In a study to be published online December 5th in Nature, the researchers report that these lowly sea creatures may provide a way to understand our own blood-forming system, improve our immune function, and find new immune-associated tools for biological discovery. The mammalian and Botrytis blood-forming system also share hundreds of homologous genes, even though the two species are separated by over 500 million years of evolution. The researchers isolated the Botrytis stem cells that are the foundation of the blood and immune system, as well as the progenitor cells they make on their way to becoming adult blood and immune cells. Out of the invertebrates, the Botrytis blood stem cells or progenitors are the most similar to vertebrate blood cells. So it is possible, if not likely, that they are the missing link between vertebrates and invertebrates. Odd characteristics. Botrytis is an organism with many odd characteristics. It lives part of its life as a free-swimming chordate tadpole, an animal without vertebrae but with a spinal bundle called notochord. Then it attaches itself to a rock, undergoes metamorphosis to lose its notochord, tail, body muscles, and one of its two brains. It settles down to live in colonies with other Botrytis organisms on the subtidal surface. Under the microscope, a Botrytis colony looks like a bouquet of flowers, although in reality each petal is a separate organism with its own heart, gills, digestive system, brain, and blood cells. The separate Botrytis organism and the colony share a common blood supply and even exchange cells. This blood sharing allows stem cells for sperm and eggs and stem cells for body tissue to be shared throughout the growing colony. The researchers showed that there are close parallels between the blood system and Botrytis and in mammals. They found that Botrytis has a sort of incubator of specialized cells called a niche that holds and supports blood stem cells and is a lot like the blood cell niche in mammalian bone marrow. They also found that Botrytis blood stem cells will find their own way from blood vessels to the niche, exactly as they do in mammals. And they found 327 genes involved in blood cell formation in Botrytis that are similar to genes involved in blood formation in mammals. Previously, researchers in the lab showed that a single variant of a gene called BHF regulated whether separate Botrytis organisms would send out blood vessels from their own bodies and merge with adjacent individuals, or undergo an immune rejection, preventing blood cell exchanges. The new study identifies how BHF regulates whether organisms fuse together in the colony. If the protein produced by the gene is recognized as compatible by the other colony, it prevents the activation of a rejection process that is similar to the way that the human immune system's natural killer cells attacks tissues that are not self. An excellent model. The discovery of such strong parallels between the two systems offers researchers an excellent model for studying many biological phenomena in mammals. The researchers said blood stem cells in mammals are hard to find, and when found it is very hard to follow what is going on in the blood stem cell niche. Botrytis is a translucent organism, so we can easily spot the niche and visually follow the migration of each type of cell from one part of their body to the other. It is also easy to observe how the cells and individual organisms interact when one mounts an immune attack against the other, while the two individuals fuse blood vessels. This could provide scientists with a better understanding of why an organism accepts or rejects foreign cells, knowledge that could give insights into an organ transplant acceptance and rejection. With its primitive but effective immune system, Botrytis may also give us insight into how we can boost our immune system responses to pathogens and cancer. But in addition to any practical benefits this research may produce, we are delighted to explore the important guideposts on the path to understand the evolution of vertebrates and of their blood-forming and immune systems. Isn't that what curiosity-driven science is supposed to do? End.